Hello students, welcome to EPG Pathshala. I am Dr. Amit Bhattacharya from Ramjas College, University of Delhi. Today we are going to discuss about a module Plasmodium drug targets part 1 under paper Zoology of parasitism. The learning objective of this module are: first, know about malaria epidemiology; second, learn about various antimalarial drug targets within the parasite; third, identify novel plasmodium drug targets or pathways; fourth, evaluate various aspects of mode of action of antimalarial; fifth, analyze vector control strategies to eliminate malaria transmission. Let's begin with the overview of what exactly malaria is. Malaria is caused by 5 species of parasite belonging to the genus Plasmodium. Of these, Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae and Plasmodium ovale are human malarial species which are spread from one person to the other person by female mosquitoes of the genus Anopheles. There are about 400 species of Anopheles mosquito, but only 30 of these are major vector of major importance. In recent years, human cases of malaria have been also recorded due to Plasmodium knowlesi, a species that causes malaria among monkeys and occurs in certain forested area of Southeast Asia. Plasmodium falciparum and Plasmodium vivax malariae pose the greatest public health challenge. Plasmodium falciparum is more prevalent on the African continent and is responsible for most deaths from malaria. Plasmodium vivax has a wider geographic distribution than Plasmodium falciparum because it can develop in Anopheles mosquito vector at lower temperature, can survive at higher altitudes and in cooler climate. Second, it also has a dormant liver stage known as hypnozoites that enable it to survive for a longer period as a potential reservoir of infection. The hypnozoites can activate months later to cause a relapse. Malaria transmission occur in all 6 WHO regions globally. An estimated 3.3 billion people are at risk of being infected with malaria and develop this disease. About 1.2 billion people are at higher risk. According to the WHO latest estimates, 198 million cases of malaria occur globally in 2013, and the disease led to about 5,84,000 deaths. The burden is heaviest in the WHO African region, where an estimated 90% of all malaria deaths occur, and in children under the age group of 5 years, who account for 78% of all deaths. The earliest known historical perspective of malaria goes back to about 4,000 years. Malaria is a disease or resembling malaria disease have been noted for more than 4,000 years, and the word malaria comes from the Italian word which means bad air. Malaria has probably influenced to a greater extent human populations and human history. The symptoms of malaria were first described in ancient Chinese writing. In 2700 BC, several characteristic symptoms of what would later be named malaria were described in Nenqing, the Canon of Medicine. Nenqing was edited by Emperor Huangdi. In 800 to 600 BC, in the Susruta, a Sanskrit medical treatise, the symptoms of malaria fever were described and attributed to bites of certain insects. In 1880, Dr. Charles Laveran discovered the parasite in the blood of malarial patient. On 20th August 1897 in Sikandrabad, Sir Ronald Ross made his landmark discovery while dissecting the stomach tissue of an Anopheles mosquito fed 4 days previously on a malarious patient. He found that the malarial parasite and went on to prove the role of Anopheles mosquito in the transmission of malaria parasites in humans. In this picture, we can see that internationally an expected 3.3 billion citizens in 97 countries and territories are at a risk of malaria, while around 1.2 billion are at high risk malaria transmission each year. In all 6 WHO regions, which includes African, regions of American, Eastern Mediterranean, European, Southeast Asia and Western Pacific have been reported with malaria transmission. The Southeast Asia region is the second most malaria affected region in the world, while Indian subcontinent in the Southeast Asian region has the highest malaria burden, followed by Indonesia and Myanmar. The malaria endemicity in India has been shown in this picture, and if we see the picture, the malaria is spread out across the Indian subcontinent. The major malaria cases are reported from Assam, Jharkhand, Odisha, Rourkela, and if we see, the major vector of malaria are Anopheles culcifacius, Anopheles stephensii and Anopheles fluentis. And the malaria species which is widespread in India is Plasmodium falciparum and Plasmodium vivax. Now, how do we identify the Anopheles female mosquitoes? The distinguishing feature of Anopheles female mosquitoes from the other mosquitoes is the palps, which are as long as the proboscis, and there is a presence of discrete blocks of black and white scales on the wings. The adult mosquito can also be identified by their typical resting position, males and females resting with their abdominals sticking in the air rather than parallel to the surface on which they are resting. There are 58 species of Indian anophelines, out of which 6 are the major vectors of malaria in different ecological settings in India. These 6 major vectors of malaria are: first, Anopheles culcifacius, a species of plain; Anopheles fluentilis, a species of forested area in central India, Himalayan foothill and in eastern India; Anopheles stephensii, a species of urban area; Anopheles dirus, a species of forested area; Anopheles minimus, a species of forest fringe areas; and Anopheles sandicus, a species of coastal area. If we see the trends in domestic and international fundings, which is being released by World Malaria Report 2014, the total annual funding for malaria control and elimination programs globally estimates to around $2.7 billion in 2013, which is about 3 times the amount spent in 2005. Funding investment has been greatest in the highest malaria burden region, which is WHO African region. So all these statistics are being shown over here, where we can see the domestic funding is less while the international funding is of a higher magnitude. Further, the growth of funding has been greatest in the World Health Organization African region, where the disease burden is greatest. Between 2005 and 2013, the international and domestic investment increased to an annual average of 22% and 4% per year respectively in the WHO African region, while in the other WHO regions it increased by 15% and 2% per year respectively. The Rollback Malaria Partnership has formulated the Global Malaria Action Plan (GMAP) and is hosted by the World Health Organization at Geneva, Switzerland. The GMAP is the leading forum for channelizing action and resources for global fight against malaria eradication. The BRIDGE program that collectively inputs of 30 malaria endemic country and region, 65 international institutions, which includes academic and research institutes, and 250 experts from various malaria field. The three core areas, which includes control efforts, elimination step, and research to develop new tools and approaches to complement global malaria control and elimination efforts. The long-term goal of GMAP is to make malaria-free world through progressive elimination step. The progressive goal-back malaria step targets to first reduce global malaria incidences by 50% in 2010 and by 75% in 2016 with respect to the number of cases in 2000. Second, decrease global malaria mortality deaths by 50% in 2010 and to nearly zero deaths in 2015 with respect to the year 2000 estimates. And third, eradicate malaria in 8 to 10 countries by 2015 and subsequently in other countries. Despite many years of research and intensive effort to control and eradicate malaria, the malaria-free world still remains a dream. The predominance of this deadly disease continues to increase in several parts of the world. In the recent era, there has been strong support for an integrated approach for malaria control, which includes the vector control strategy that reduces the transmission of mosquito vectors from humans to mosquitoes and then back to mosquitoes. Second, insecticide-treated mosquito nets. Third, indoor residual spray. And fourth, chemoprevention, which prevents the blood stages infection growing in the human blood circulation. And finally, it also includes case management where proper diagnosis and treatment of the infections are done. Existing strategy of malaria control in India broadly includes first, diagnosis, which includes microscopic examination of the blood smear, rapid diagnostic tests which are available in the market. Second, the treatment regimes which are being quantified by national drug policy, which includes the various anti-malarial drugs. And third, vector control strategy, which includes larval control, indoor residual spray, and insecticide treated nets. All these broad areas of control strategies have been shown in this picture. The vector control strategies to control and eliminate malaria vector can be broadly classified into three main categories. A. Reducing vector density. B. Interrupting the vector life cycle. C. Preventing the mosquito bite, that is by creating a barrier between the two hosts, which is the mosquito and the humans. Further, malaria vector control measures have been shown in this pictorial representation, which includes environmental source reduction, chemical and biological larvicide protection or control, human vector contact prevention, outdoor insecticide spraying, and indoor residual spraying. So all these forms a major part of malaria vector control strategies. In malaria programs, the vector control measures include interventions that restrict the transmission of malaria parasites by controlling the vector population. As the female Anopheles mosquito is a moving target, hence the vector control measures can be broadly classified into various categories which we discussed. Over here in this pictorial representation, we can see the various integrated vector management strategies which are being used to control the vector-borne diseases. These includes indoor residual spray, B. insecticide treated nets, C. space spraying, and D. chemical larvicides and biolarvicide being sprayed into the local areas. The vector control strategies, according to ICAMR, includes various different ways. In India, malaria is transmitted by 9 vector species, of which Anopheles culcifacius and Anopheles fluviatilis are responsible for 60 to 70% and 50% of new malaria cases respectively. For adult vector control, indoor residual spray includes various wettable powder formulation of different insecticide such as DDT, which is dichlorodiphenyl trichloroethane 50% WP, malathion 25% WP, and synthetic pyrethroids 25% WP. The synthetic pyrethroids insecticide contains deltamethrin 2.5% WP, cyphlothrin 10% WP, lambda cyclothrin 10% WP, and alpha cypermethrin 5% WP. The larval control spray, which is also called as the emulsion concentrate, uses Temphos 50% EC and Fenthion 82.5% EC. The insecticide impregnated bed nets are also treated with these synthetic pyrethroid insecticides and have proved successful in the vector control measures. Insecticide-treated bed nets are estimated to reduce malaria mortality rates by 55% in children under the age of 5 years in sub-Saharan Africa. This is as per the latest World Malaria Report, which is being released by World Health Organization. The insecticide-treated bed nets have been shown to reduce incidences of malaria cases by 50% in a variety of settings. When the insecticide-treated bed nets are used by pregnant women, they are also efficacious in reducing maternal anemia, placental infections, and low birth weight. The other strategy to control and eliminate malaria is chemoprevention by using antimalarial drugs. Subsequently, the understanding of parasite development process in mosquitoes further led to several new strategies in vector control in the earlier 20th century. In the early 1600s, decoction of cinchona bark have been reported to be effective healer for malaria fever and have been used extensively for centuries. Over the decades, relevant progress in malaria drug discovery has been made to extend antimalarial arsenals. These drugs, which have been shown in this figure, majorly target the blood stages to relieve the symptoms, the liver stages to prevent relapses, or the transmission stages to protect further from any widespread. Newer blood stage targets are being discovered, which are being prompting to have multi-target approach for inclusive parasite clearance from the blood circulation. The drug discovery specially aimed at hepatic and transmission stages are crucial, as targeting these vital stages could be instrumental in eradicating malaria. So in this picture, if we see, the various antimalarial drugs have been shown, which includes quinine, chloroquine, amidoquine, primaquine, mefloquine, artemisinin and its derivative, which includes artesunate, dihydroartemisinin, artemether and arteether. The timeline shown over here shows the various developmental processes of the discovery of antimalarial drugs. For decades, there were no precise malaria treatment until in 17th century, when the Spanish colonizer carried back from Peru cinchona tree bark, primarily referred as the juiciest bark or sacred bark, which was later on called as quinine and was extracted. Consequently, researchers identified quinine, an active ingredient isolated from the cinchona bark, in the year 1820, and it soon became the first recognized anti-malarial drug. In 1856, English chemist William Perkins tried to synthesize quinine but failed in his attempt. In the subsequent years, Paul Ehrlich, a noted German scientist, documented that methylene blue, a synthetic stain which is being used to stain these slides, was predominantly useful in staining malaria parasite, and hypothesized that if the parasites on the blood smear slide took up methylene blue dye, then it might kill it also in vivo. And subsequently, he went on to prove methylene blue as one of the first synthetic antimalarial drug used for human patients. Several failed attempts to chemically synthesize quinine were made following years, but German chemists pioneered to produce new synthetic antimalarial drugs based on quinine structure in 1944. Of these synthetic compounds, chloroquine became one of the most potent and well-known antimalarial drugs until 1977, when these drugs collapsed to the emergence of chloroquine-resistant parasites. Over the years, several antimalarial drugs have been developed to treat and delay the menace of growing resistant parasites. Of these include the plant-derived artemisinin derivatives, a age-old Chinese herbal remedy which has been documented and now proven as the first-line therapy of malaria. So now artemisinin combination therapies are being recommended by World Health Organization for the treatment of uncomplicated as well as complicated Plasmodium falciparum and other malaria cases. The antimalarial drugs are classified into various groups. Of these, the 12 important groups are: first, cinchona alkaloid, which includes quinine and quinidine; second, 4-aminoquinolines, which include chloroquine, amidoquine, and piperacin; third, diaminopyrimidines, which includes pyrimethamine; fourth, 8-aminoquinolines, which includes primaquine; fifth, sulfonamides and sulfone, which includes sulfadoxine; sixth, sesquiterpene lactone, which includes artesunate, artemether, arteether, the various derivatives of artemisinin; seventh, quinone methanol, which includes mefloquine; eighth, tetracyclines, which includes tetracycline, doxycycline; ninth, amino alcohols, which includes halofrantine and lumefantrine; 10th, Munich bases, which includes pyronazine; 11th, naphthoquinone, which includes atorvaquone; and 12th, biguanides, which includes proguanine and chlorproguanine. The picture over here shows the life cycle of malaria parasite and the anti-plasmodial drug targets within the human host, which is going under the asexual development of the parasite. So if we see the picture over here, there is a liver schizogony and the erythrocyte schizogony which is going on. So many of the drugs like primaquine attacks these liver schizon stages, which are many times known as the hypnozoites, while number of drugs like quinine, chloroquine, mefloquine, sulfadoxine, pyrimethamine and artemisinin targets the intracellular erythrocytic stages which grow and develop within the human RBCs, while some of the drugs like primaquine and chloroquine targets the gametocytes which are being formed and subsequently taken up by the mosquito for the second round of infection in a new host. The first widely known remedy of malaria fever was discovered in Peru and Ecuador. It was the bark of the cinchona tree, a close cousin of coffee. Local people called the remedy quina quina, which means bark of barks, and it was later distributed worldwide as quinine. Quinine, a constituent of the cinchona tree bark, was used by Jesuit missionaries in South America to cure malaria from as early as the 1600s. In 1820, Pelletier, along with another French chemist, Caventou, isolated quinine from the bark of a cinchona tree for the first time. Pelletier has initially kept his discovery of quinine quiet, but once he has published his secret extracted quinine, it quickly became and established itself in the medical practice. As in the year 1820, Pelletier and Caventou extracted and isolated the quinine from the bark. Subsequently, purified quinine then replaced the crude extracts of the bark as the standard antimalarial treatment used for malaria fever. Soon, it became widespread and was recommended as a potent antimalarial drug. Cinchona alkaloid, which is quinine, belongs to the aryl amino alcohol group of drugs. Various formulations of quinine salts are available, such as hydrochloride, dihydrochloride, sulfate, bisulfate, and gluconate salts, while the dihydrochloride salt is the most extensively used of them. Quinine is a rapid and potent sysonticidal antimalarial drugs against intraerythrocytic stages. It is also shown gametocytocidal activity against Plasmodium vivax and malaria species, but not against Plasmodium falciparum gametocyte. However, the exact antimalarial inhibitory mechanisms of quinine remains elusive. Quinine has been also showing mild antipyretic and analgesic activity. The next drug which was discovered was called as chloroquine. The supply of quinine throughout the world was disrupted as the Japanese army took over the Java during the World War II. In 1920s, 8-aminoquinone plasmoquine, the precursor of plasmoquine, and mepacrine were manufactured by researchers in the Bayer laboratories. During the Second World War period, the American, British, and Australian scientists collaborated in a significant attempt to develop new synthetic antimalarials. With their cumulative effort, about 16,000 candidate compounds were synthesized and subsequently tested. Surprisingly, associated scientists informed about resocin developed in America as chloroquine and sontocin. Resocin, which had a acquisition number of SN183, was tested, but due to high toxicity profile, it was not considered further. By 1946, US clinical trials subsequently reported that chloroquine, which was renumbered as SN7618, as a powerful antimalarial than other synthetic forms. So if we see the picture over here, we can see a chloroquine tablets which is of 250 mg shown over here. The various formulations of chloroquines are available in the market. These include chloroquine phosphate tablets, chloroquine phosphate injections, and chloroquine syrup. So depending upon the intake capacity of the patient, the doctor recommends and prescribes these particular formulations accordingly. Antimalarial property of chloroquine, which is shown in the figure, and quinine is mainly because they interfere with the essential hemoglobin degradation pathway of the parasite. The hemoglobin acts as a major food source for the parasite because it takes up the globin or the protein part, while the heme part is subsequently polymerized to form the hemozoin fragment. Non-toxic beta-hematin is called as the hemozoin. Chloroquine averts the neutralization of this toxic ferriprotoporphyrin 9 in the parasite. The heme polymerase enzyme, which is located in the acidic food vacuole of the intraerythrocytic parasite, utilizes this ferriprotoporphyrin 9, which is released from the hemoglobin catalysis, as the substrate for hemozoin biosynthesis. If we see the picture over here, the heme which is released from the hemoglobin catabolism is being polymerized with the help of the heme polymerase enzyme, which is located in the acidic food vacuole of the intraerythrocytic parasite. So this heme is polymerized to form the hemozoin fragments. So this hemozoin fragment is non-toxic, while the heme part was toxic to the parasite. So it is converting the toxic heme into non-toxic hemozoin. So what happens is, as soon as the chloroquine is given to the patient, this chloroquine gets into this acidic food vacuole and it disrupts the neutralization of this toxic ferriprotoporphyrin or heme in the parasite. The hemoglobin is being utilized by the parasite to take up the globin part, and the toxic heme, which is called as the ferriprotoporphyrin 9, is polymerized to non-toxic beta-hematin and stocked in the food vacuole, a protective mechanism developed by the parasite against its self-destruction. It has been reported that non-polymerized ferriprotoporphyrin 9 is highly toxic to the parasite as it damages the proteases and disrupts its cell membrane. So this ferriprotoporphyrin can also accumulate by binding with the heme binder and subsequently goes on to form the hemozoin. But as soon as the chloroquine accumulates into the food vacuole, it competes with the heme binder and binds to this toxic ferriprotoporphyrin, causing the destruction of the parasite, because these unpolymerized heme or the ferriprotoporphyrin are toxic to the parasite, causing the damage to its cell membrane and the various enzyme system. So students, let us summarize what we have learned in this module. Malaria parasite, a hematoprotozoan parasite transmitted by certain Anopheles mosquito species, have a complex life cycle. Malaria can have devastating consequences in pregnant women and in children. The World Health Organization vision is for a world free of malaria that can be achieved through elimination of malaria infection followed by global malaria eradication programs. Malaria intervention comprises of, first, vector control strategies, which reduces transmission by the Anopheles mosquito from humans to other mosquitoes and then back to human host. Second, insecticide treated mosquito nets. Third, indoor residual spraying. And fourth, chemoprevention by antimalarial drugs. In areas of high malaria transmission, WHO recommends the use of chemoprevention strategies. The three potential roles for the antimalarial medicines are prophylactic, therapeutic, and transmission blocking. Over the decades, the malaria chemotherapy has relied largely on antimalarials such as quinine, chloroquine, sulfadoxine pyrimethamine, mefloquine, and to the latest one, the artemisinin combination therapies. They have been so successful as they are inexpensive and widely available. So with this note, I conclude my lecture. Thank you very much.